Welcome, everyone, to the April meeting of the Caribbean Cooperation Team, or CCT. We are just so excited today that we can showcase with you, share with you, the new Caribbean coral restoration efforts that are being funded by the CORDAP program. We have a ambitious agenda for today. I confess I lost it about half an hour ago, and it hasn't yet made its way into the program that you're seeing, but we'll be sure to send it out afterwards if it doesn't turn up at the end. And I will just plunge straight into the background, and I should put the agendas in the sections. Okay. We'll find the agendas when we get there, and we'll start with the background. And I want to express my appreciation for the financial support from AGRA, MPA Connect, the Ocean Research Education Foundation, and now also CORDAP. And I thank all the peoples of the Caribbean who are working to rescue corals, repair reef habitats, and improve the health of the oceans and its creatures. Next, please. In 2018, when stony coral disease had spread from Florida to the first 3 of what are now 30 jurisdictions across the Caribbean, NOAA's Dana Wissenich-Mendez invited me to be her co-host for a virtual group that's now called the CCT. It has since evolved into a regional information exchange for also responding to stressors affecting coral reefs. In addition to stony coral tissue loss disease, we've experienced the 2022 die-off of diadema, the huge mass bleaching event last year, and now eunomia and possibly other Indo-Pacific soft corals that are behaving invasively in the Caribbean. Next. By 2022, SCTLD had seriously wounded or killed so many Caribbean corals that the CCTs pivoted from sharing educational and training outreach efforts like shown here to actively promoting the region-wide preservation of our precious, vulnerable, and absolutely unique endemic stony corals. Colleagues have virtually shared their relatively low-cost approaches for propagating corals or herbivorous urchins with those of our partners who are seeking funds to scale up their own local research efforts. Thanks to CORDAP, we can now scale up our regional offers, as AGRA's Patricia Kramer will soon explain. But first, next. Oh, we were supposed to be there already. I thought something was wrong. We will now scale up our regional offerings, as AGRA's Patricia Kramer will soon explain. But first, we're going to hear from CORDAP Program Manager Anderson Mayfield. Next. Oh, wait a minute, back up. Sorry, folks, we didn't, because I lost these slides, we didn't practice beforehand. First, Anderson Mayfield will give a brief introduction to the CORDAP program, and then in part 1, we will hear the overviews of the 5 projects that CORDAP has funded for work that will be exclusively in or will include the Caribbean in this coming granting period. And they will be provided by the people whose names you see here, and you'll get to meet them as the program continues. And then the last of these will be Patricia Kramer describing our own CORDAP program. So if we could have the next slide now, Anderson Cooper, the CORDAP program manager, will give us a short overview of this exciting new program. So please welcome him. And Anderson, take it away.
All right. Well, I hope you guys can hear me okay. I'm actually sitting outside. I'm supposed to be in another meeting right now in Barcelona, but I was really excited about the promise of talking to you guys a little bit about CORDAP. And I think, seeing the list of attendees, I know a lot of you guys from my past life as a coral researcher, and also definitely familiar with those of you who are already funded in our first round of projects. So a lot of this is going to be a little bit of preaching to the choir and things that a lot of the audience will already know, but for those of you who are not as familiar with the CORDAP and what we're trying to do, hopefully you'll learn something here in the next few minutes. So next slide, please. So the CORDAP stands for the Coral Research and Development Accelerator Platform. It was a G20 initiative that began a couple of years ago when Saudi Arabia held the G20 presidency. And really our overarching objective is this bullet point A, funding and delivering the best novel scientific and technological solutions to conserve corals and restore coral reefs. So in the next few minutes, I'm going to talk about how we do that through a program that we call the Coral Accelerator Program. But you can see we have all these other lofty goals and objectives. And I think, we're a relatively small team and we've been busy and we've been chipping away at some of these, but maybe not as much as we should have. So if you look at, let's see, for instance, we've got some of these that deal with capacity development. This is something that we really started to prioritize in particular this year, knowing that the majority of coral reefs are found in the global south. So, you might have a great technology that works, in a place like Miami or where I was living before, where, you've got a significant amount of financial resources and it might, be able to save a couple of corals. But if it's not something that could be deployed at scale at a relatively affordable, level, it's not going to be something that CORDAP is probably going to be that excited about. And this is something I struggled with in my own research. We would have something that would have been really good for science, good for your CV, good for knowledge, but maybe it was something that was too expensive or too impractical to save corals. So this is something we're definitely looking at in the decisions for what we want to fund. Another thing that we're just kicking off in collaboration with the Coral Restoration Consortium is this point G, and this is the developing and sharing novel scientific tools. There's definitely already some great online source material out there for things like restoration, and there's obviously the peer-reviewed literature, but we really want to invest in having some kind of truly centralized hubs for practitioners to go to for protocols, for data, for anything, starting with potentially very little knowledge to be able to plan a full-fledged, reef intervention. I think this is something that's really attractive to me, and it's something I hope we'll be able to make progress in pretty quickly. Next slide, please. Oh, yes, a couple of these you could skip. Okay, so I already mentioned kind of our core initiative, this Coral Accelerator Program. So we had our first call in 2022. So sounds like we're going to hear from 4 or 5 of the 14 funded projects over the next hour or so, which is awesome. We had $18 million to give away, and we spread this out over 14 projects that spanned from 1 to 3 years, with most being the full 3 years. I'm going to talk a little bit more about the details of this Coral Accelerator Program in another slide. But right now, we're pretty much right in the middle of the second call, the CAP 2023. So we started with about 130 proposals. The top 36, 37 made it to the next round, and we'll probably fund, a similar number, so 13, 14. So about a third that made the cut to the next round are going to be supported. We also are not just exclusively doing the funding program and these capacity development initiatives and the data sharing. We're also hosting our own workshops and what they call scoping studies and roadmaps. And these are areas where we realize it's important for coral conservation and restoration, but maybe more study and planning are required before the funding will be effective. So these are basically kind of brainstorming focused workshops, and we've had some to date on assisted evolution, coral aquaculture, cold water corals, ecological risk, and then the capacity development one that just wrapped up a couple of weeks ago. So these are not only going to be kind of where we see as critical areas for future funding, but, as, if not more importantly, the publications and the roadmaps to come out of them we really hope are going to be valuable resources to the field or for people that are interested in leveraging kind of the science behind these topics in their own coral conservation and restoration works. And as I mentioned, capacity development is fully on our radar now. It's something we always realized was important, but we didn't really have much of a chance to go into it in much detail with a huge amount of effort until this year. So I hope you're going to hear some more good news about what we're doing in this field soon. And now we have a dedicated fundraising team, because right now it's a G20 initiative, but all of our funding is coming from the government of the Kingdom of Saudi Arabia. The other G20 countries haven't yet chipped in, but we're optimistic. And we've started a foundation to where we can actually accept funds from outside the kingdom, which wasn't actually possible before. So, philanthropy, private sector, these are all sources of funding that we want to tap into to get the money to you guys. And it's important that the university we're based at, King Abdullah University of Science and Technology, pays all of our staff salaries. They pay for our accommodation, for everything. So what that means is, none of these funds going into CORDAP, there's no overhead being taken out. 100% of it can go to science, which is a really nice perk, I think. Next slide, please. So I mentioned I would give you a little bit more detail about the nature of the Coral Accelerator Program for those of you who aren't already awardees or applicants. These are internationally collaborative teams. You have to have at least 2 countries involved, with at least 3 applicants. And it's very important that one of these countries must be a low or middle income country. So we're not going to fund a project between the United States and Australia, for instance, even though we know, it would be scientifically very fruitful. This is not, our sole focus. We're trying to fund excellent science, but also make sure the technology and solutions are getting to where they're actually needed. So you're going to have at least one developing country on your project. Where you're at in terms of how far along your project is it at the pilot stage almost? Are you just about to go live with your final field testing? We'll fund anywhere in that process. Right now, I mentioned we're giving out about 18 million US dollars a year. So the projects are capped at about 1.5 million per project. So that's why you get to that 13, 14 projects per year number. And they can run between 1 to 3 years, with, again, majority of people choosing the longer side, because we know, for most of these projects to see impact, even 3 years might be a relatively short project, depending on what you want to do. So we have a 2-stage review. We have a really excellent scientific advisory council, of which a few of them I think are actually on the call. So they'd also be good resources to tap into about suggestions and tips. But basically we rely upon them to kind of whittle down our long list of these pre-proposals into the ones that get invited to full proposals. And then those that get invited to full proposals, we go into more of a external review that's more akin to having, your scientific manuscript reviewed. We're going all over the world trying to find expertise that can best give constructive criticism on these proposals. So this is a pretty lengthy process. So people are submitting their proposals in November, hoping to get answers by June. And one important thing is, and I think it should probably not be a surprise, is our very strict kind of IP policy. We know that some of these projects are going to be coming in with background IP, and there could be some sensitive issues there. But just like I was saying, we won't have done our job if we only funded Global North projects. If we're only funding projects that have proprietary data or technologies, or basically that are going to drive up prices to use them, this is not going to be acceptable for us. If you need to cover your expenses and your own costs for the technology, that's okay. But we're not trying to have people get rich off these CORDAP projects. These are meant to be, affordable solutions that are getting out there to where they're needed most. This is open access, preferably free, or at least affordable. This is very important for us. And, because and it's important to mention too, because we do allow industry to apply, which is uncommon for funding agencies, but we require that they invest to the point where they're no longer making a profit. So we encourage industrial collaborations, but there's some restrictions because we want Jason to be as if they can slide, please. Uh-oh, I'm getting internet warnings. Can you? Oh, there we go. I'm not going to go into this in too much detail, but I mentioned earlier that our series of scoping studies kind of fuel priority areas. So the list of priority areas essentially reads as our list of scoping studies, priority areas that, we would of course always want to have. R&D capacity building, that's always going to be there. This is never not going to be a priority for CORDAP. Developing country R&D methods. Again, this is not going to be something we ever say, oh, we decided this is not a priority anymore. So essentially this list is only going to get longer. That's to put it briefly. And I didn't mention this earlier. So we had about 120 eligible proposals this year requesting in total about 160 million US dollars, of which we can give out 18. So I look at this as being, this is kind of what pains me, because $18 million to go to conserve coral reefs is a momentous achievement that I'm really proud of, but that's 11% of our overall ask. And, who knows what fraction of a percent of what we actually need. And this is why the fundraising side of things is going to be so important. Next slide, please. Oh, here
Have
we go.
we lost Anderson?
No, I had a little bit of a delay, but I'm back. I'm just going to leave you with this. This is kind of our upcoming events and initiatives over the next year or so. So as you all are well familiar, coral disease is horrifying at the moment and has been the last few years. This is certainly something that, we have disease projects already being supported, but it's something where we feel like we're still not doing enough. So we're going to host a workshop on this in Puerto Morelos later this year. We're going to have our third call coming out in November. It's going to be another 18 million US dollars, with disease likely to be, or surely to be, a priority area. The second new priority area, which has always kind of been there, we never really pitched it as such, is this idea of coral rescue, things you could do to essentially come to the aid of a reef that might be, catastrophically. So this has not really been fleshed out very well yet in terms of the schedule and logistics, but it's probably going to be sometime in late 2024, early 2025. And this, I don't want to take up too much more of your time, but this is something I'm kind of, struggling with is, if we have a 7 or 8 month review process and somebody has a coral emergency, well, that's not really commensurate with the timeline in which you need those funds. And, our kind of answer at the moment is, well, somebody else should fund those kind of coral emergency situations. But I'm completely given up on the idea that we might be able to have some kind of, I don't know, short-term, smaller, easier to access pots of money. And these are the kind of conversations we can have with our governing board each year and say, hey, maybe we can earmark some funds for these kind of things, because it's something that I think we might have a role to play in this. And then, the final thing that I'm excited about, that we're really just kind of discussing this week for the first time, is we want to get more involved in coral conservation, restoration, education. So, I mean, we don't need to reinvent the wheel in a lot of places. There's some excellent online material, from what is a coral all the way up to, restoration methods. So it could just be a matter of facilitating and coordinating a lot of these things. We definitely want to have, if some of you might remember the Poly program that they had at the Hawaii Institute of Marine Biology a long time ago. It was just this multi-month gathering of typically early career people to come and do projects, learn from each other, and it was great. And I met so many of my colleagues there, and I think the funds dried up about 15 years ago. So I think having something like this kind of resurrected would be really cool, like a multi-week, multi-month training, hands-on training at possibly where we're based, or although it could be held locally as well. I think we have a lot of potential for doing these kinds of education from the college level all the way up, and practitioners and whatnot. But it's really kind of a work in progress, and we've got a lot of different ideas. So definitely, if you say, hey, if you were going to invest in one kind of educational component for CoreDAP, you should do this, then definitely reach out to me via email. We definitely want to hear from all of you what you think would be most useful, because I think we could really do some good here too. Yeah, and that's basically all I wanted to say to you guys today. But yeah, it's my pleasure working with all of you, and I'm sure those of you who don't already have CoreDAP funding, I see a lot of people who've submitted this year, and hopefully we'll be in touch in the future with what CoreDAP can do for you. So yeah, thanks, and look forward to working with you all.
Anderson, thank you ever so much. If we could advance the slides. We have a really tight agenda today, so we're going to switch seamlessly into the talk about upscaling and optimizing coral sexual propagation technologies for coral restoration in the Caribbean that Maria Villalpando is going to present for us. And I put in the chat, please put your questions here in the chat, and we will address them at the end if time is available. And if not, we promise to answer them afterwards. So Maria, take it away. And I hope you can stay.
Thank you to the CCT. Thank you, Judith and Patricia, for spearheading this networking effort to collaborate between the Caribbean CoreDAP partners and amplify our joint efforts. We think it's highly important and relevant. I am Maria Villalpando. I'm the Research and Development Manager for Fundemar in the Dominican Republic, and I'll be speaking on behalf of Rita Sellares, our CEO, who is currently on the field, and as well as our project partners, SEACOR International and Reef Renewal Foundation Bonaire. Next slide, please. So first, before we go into the task and the activities, the overall project aim is to improve novel coral larval propagation technologies to optimize efficiency and production, maximizing coral recruit outputs with lower mortality rates, while lowering the time and resource investments in 3 key stages in gamete collection, settlement, and post-seeding survival. Fundemar in the Dominican Republic and Reef Renewal Foundation in Bonaire, who both have successful assisted sexual reproduction programs, will be implementing, testing, and enhancing these technologies developed by SEACOR International, to establish and replicate protocols in other Caribbean regions and possibly beyond. For the first task, we want to, like I said, implement and test these technologies in the DR and Bonaire. For the first technology, we used to have multi-colony collectors for nurseries, but after the massive bleaching event of last year, our nurseries most likely won't be able to recover in time for multi-colony collection. So we switched gears into refining and massively produce individual gamete collectors. This is a strategy that we used last year, since now colonies are more scattered due to stony coral tissue loss and bleaching. We are increasing our gamete collectors. Last year, we used 60 nets at the same time to collect from Diplorella labyrintiformis, and it worked really well by having collaborators, dive centers joining us for the collection efforts. And this year, we're aiming at having 80 nets for collection, knowing that we are right now in a critical situation, still with having coral still paling and the temperatures rising very fast. The next technology developed by SEACOR would be novel settlement substrates. Hopefully, we want to increase retention at least by 25% with these new substrates, increase the handling capacity, having over 25 units handled at the same time by one diver, and reducing the unit production cost to less than $1. We also want to scale up the existing technology. So the coral rearing in situ basins, and that a lot of you know, the cribs, the goal is to optimize the space and increase substrate capacity by 50% than we currently have. Now SEACOR is working on a new suspension methodology that will be tested this year in Bonaire. and seeing like what else we can do to optimize the space already available in the cribs. We also want to integrate early live feeds with Artemia, which has been shown to increase post-seeding recruit survivorship by 50%, and integrate this in our laboratory, in the cribs, and also in larval in situ tents. We also will be testing the surface deployment of substrates, the conventional C-core substrates and the novel substrates, hopefully being able to decrease outplanting labor by 50% and maintaining current retention, substrate retention rates that are around 40% in the Caribbean region, depending on the site, as well as maintaining survivorship rates that we currently have. And for the last technology, we used to have testing direct seeding plots, but we saw that the results were highly variable and it required a high volume of embryos that we think we will not be able to guarantee due to the context that we are at now. So we switched towards implementing larval seeding tents, try to make them easily replicable, also because they are most likely less affected by high temperatures since they would be on the reef floor instead of floating as the cribs, and protected from storm impacts as well. And we know these have been successful efforts with other partners in Saint-Croix and Curacao. We're also testing this in the DR and in Bonaire. Next slide, please. The second task is to expand Fundemar's land-based larval breeding facility in the Dominican Republic. Our goal is to scale up production by doubling the seeding units produced and reproducing 1 to 2 more coral species. According to C-Core, Fundemar is one of the organizations in the Caribbean with the highest recruit production rates now. We only have 12 aquariums and we have used 5 cribs at the same time before. So hopefully with this new lab that will be ready by this summer, we'll have 40 aquariums for coral recruit production. So yes, the goal is to double the numbers that we're currently doing. Doubling substrate holding capacity to also have as a safeguard ex situ, along with the cribs in situ, and host recruits for a longer period of time, as well as incubate the early live feeds for settlers. In Bonaire, Reef Renewal Foundation will formally plan and design a laboratory based on what has been working in the DR and adapt it to their local context for this project. Next slide, please. So this laboratory will be part of the new facilities of Fundemar in Bayaibe in the Dominican Republic. That's part of the DR's Marine Innovation Hub in collaboration with Grupo Fundación Punta Cana. And we will not only have the recruit production lab, but also we will have 2 other labs for experiment and testing. We will have co-working spaces and dormitories for associated researchers that would like to come to the DR to test their technologies. Next slide, please. And the little location spot is where the facilities will be, and our backyard is right there, our main restoration site, along with where we put our cribs. Usually water is conditions are very well in Bayaibe to test throughout the year all of these technologies, and hopefully we'll be able to scale up starting this summer all of the coral recruit propagation efforts. Next slide, please. For the third task of the project, we will do a technology and knowledge transfer between Fundemar and Reef Renewal Foundation. It will be focused on the coral reproduction approaches, asexual and sexual, that each institution implements, as well as the active collaboration in the implementation of the novel technology developed by C-Core in their own country. We will learn from each other from all of these efforts, try to integrate what works best for each of us. And we think that this exchange is highly important to secure the success of our programs, as well as learn about how each organization has been sustainable through time, through community integration, and have feedback throughout all the project period to refine all of the technologies. Next slide, please. For the last task, we want to communicate results with experts. We hope to have by the end of the project period protocols established for the technologies that have been successful. as well as a documentary style video and video material for training other partners in the Caribbean on implementing this new technology. We will be hosting a training workshop in the Dominican Republic with partners from Honduras, Colombia, Costa Rica, and Cuba, as well as hosting an online webinar for the wider larval propagation community, and present project results at scientific conferences such as Reef Futures, which we know that a lot of you will be there. So we're very happy to meet you all, hopefully in person, and keep talking about all of these efforts and how we can collaborate with each other and learn from each other to upscale the much needed coral restoration efforts in the Caribbean. Thank you so much. That's all from my part. And if you have any questions, I'll be around.
Thank you so much, Maria. Our next speaker will be Andrea Godoy Mendoza, and she will talk about interventions to improve the health, growth, and survival of larvae and settlers for sexual reproduction of the Mesoamerican reef. Andrea.
Thank you so much, Judy, and everyone for joining. Good morning. My name is Andrea Godoy Mendoza, and I am Program Manager for Roatan Marine Park. And I am very proud to be presenting today on behalf of the co-principal investigator team from California Academy of Sciences and Roatan Marine Park that I am a part of. As Judy mentioned, the name of our project is Interventions to Improve the Health, Growth, and Survival of Larvae and Settlers for Sexual Restoration of the Mesoamerican Reef. Next slide, please. As part of our co-principal investigator team, we have our lead PI, Dr. Rebecca Albright. She is curator of invertebrate zoology and also the founder of the Coral Regeneration Lab from California Academy of Sciences. Also, Dr. Elora Lopez Nandam, who is a postdoctoral researcher who has been leading the experiments and interventions that I will be describing in this presentation under Dr. Albright's supervision. Joining me in the Roatan Marine Park co-PI team, we have Jennifer Keck. She is Education and Research Coordinator at the Roatan Institute for Marine Science, and she is also a member of the Board of Directors of Roatan Marine Park. Next slide, please. The main objectives of our project is first to increase local capacity for coral restoration through the training of partners, starting with Roatan Marine Park, but with the intention to hopefully expand to other partners in the broader Mesoamerican Reef region and also Central America. This in order to develop and implement coral husbandry and sexual reproduction techniques. Also, another one that we're very excited about is design and build a larval rearing and experimentation facility, which will be the first of its kind in the country, potentially even Central America. And we aim to make all of the designs, budgets, protocols freely available in both English and Spanish for anyone that's interested in the region or at a broader level. And last but not least, within this 36-month project, we will also be testing low-cost and low-tech interventions in order to try to increase growth and survival rates of coral settlers or recruits, and to also be able to select for heat-tolerant individuals. All of these will later be implemented directly in the field, starting in Roatan and also potentially and hopefully to a broader level. Next slide, please. Now, these 4 interventions consist, first of all, of seawater buffering. Basically, we will be increasing water alkalinity in order to enhance that skeletogenesis at the very early stages of coral development. We will also be doing amino acid dosing, and this occurs during larval culturing. We will be giving larvae amino acids in order to help them nutritionally and to aid in their growth and development. Also, during larval culturing, we will be doing symbiont addition, where we basically inoculate the larvae with a symbiont, and this will also aid in that energy needs and be the source for energy in these early stages where there's a very high energy demand. And last but not least, we will also be doing an experiment for heat selection, where we're basically trying to select for thermally tolerant individuals at this very early life stage in order to try to speed up adaptation to higher temperatures, which is what we are seeing all over the world right now. And this, we hope to observe it within the surviving population. Some of our initial results or preliminary results from the ex situ spawns that we have conducted with Acropora millipora, which is actually a Pacific species, have yielded some potentially promising results. Next, please. Now, this basically consists of batch fertilizing all of the gametes from the different spawners that the California Academy of Sciences has within their facilities. Then we split them into control groups and into all of the treatments that I have previously described, the amino acids, the symbiont, and the buffered water. And then these are submitted to very similar common conditions, and we proceed to have the larvae settle into tiles that are very similar to what is used in some microfragmentation approaches. And then we monitor for anywhere between 6 to 12 months for differences in growth and survival. And then they're also submitted to the heat tolerance experiment. As I mentioned earlier, our preliminary results from the initial experiments that were conducted at California in January of 2022 and December 2023 suggest that we might be able to increase those or to actually decrease those bottlenecks that occur post-settlement and also when it's already at their development stage. Next, please. Now, a little bit of the progress that we have made so far. Back in November and December of 2023, a team of 3 members from Roatan Marine Park visited the California Academy of Sciences facilities, and we spent close to 4 weeks facilitating the Acropora millipora ex situ spawn. We were learning directly under Rebecca Albright and Dr. Elora Lopez-Nandam for these spawning events. We learned about husbandry, how to conduct these interventions, how to perform them, how to actually maintain them for longer periods of time. And since then, all of the settlers that I mentioned earlier are now on tiles, have been photographed, and we will continue to monitor them periodically in order to see what the recruit survival and growth is, and that will continue for the next at least 12 months. Also, another important milestone is that since we received the funds in February 2024, we are now finalizing the details for coral larval rearing and experimentation facility that will be built in Roatan, very close to where our offices are right now, actually in our very backyard, and something that we are very excited about, and where we also hope to be able to host researchers and people that just want to help out with our conservation efforts. Next slide, please. Some of the upcoming activities that we have planned, the facility that I mentioned, we really hope to be able to have that ready by July, but with some of the delays that we have been facing, we expect that there might be a slight delay, but that is in the books now. We will also be implementing all of the interventions that yielded the most successful results in Roatan with our Acropora spawn that is meant to happen in August. And then in December as well, California Academy of Sciences will also be refining testing of the most successful interventions in December, later this year. Now, another thing that we have to really get through is the bottleneck of analyzing all of the images that we have generated. And we are in the process of trying to make that a bit more efficient in order to be able to analyze all of the results that we have had in the past few months. Next slide, please. And here is just some information about our partners. We're partnered up with the California Academy of Sciences, and I am part of Roatan Marine Park. Here is just some information. And if you would like to know more, next slide, please. Here we have our COPI team contact information, and if there are any questions, I would be happy to answer them later on, or if you want to reach out by email, I am also available. Thank you very much.
Thank you, Andrea. So far, I've been just so amazed by all the new efforts that are being planned with these grants that have been recently awarded. And for more of this, I'm sure, I now introduce Valeria Pizarro, who will be talking about giving Caribbean corals a future, stony coral tissue loss disease, and the use of probiotics in coral restoration projects. Valeria, welcome.
Thank you, Judy. Well, thanks to you and Patricia and the Agro team for inviting us for this talk. I think that is amazing, and especially hearing all the projects that we are having in the Caribbean, as you said. So, and of course, to CORDAP for the funding, that this is really important for us. So our project, it's a collaboration between 3 Colombian institutions, Coralina, that is environmental agency that or the environmental entity, the public environmental entity that works the archipelago of San Andres, Providence, and Santa Catalina. We have the Ecomares, that is a local NGO that works mainly on larval propagation projects, especially for this project. Blue Indigo Foundation, that is a local NGO that works in San Andres. That is where we're going to have the project that is going to be leading the microfragmentation. And we have 2 US universities, the University of Massachusetts Lowell with Dr. Sarah Knox Wolfson, who will be leading the metagenomics part of this project, and the University of North Carolina, Wilmington, with Dr. Blake Ushijima, who will be leading the probiotics part. And the Pear Institute for Marine Science that I'm part of, that will be we are leading the project, and we will be focusing on stony coral tissue loss disease, like the assessments, phytomosaics, and of course, collaborating with everyone. Next. So this project, actually, when we started talking about it, I was dealing with the stony coral tissue loss disease in the Bahamas, and then the disease started to spread to the archipelago of San Andres, Providence, and Santa Catalina, that belongs to Colombia, in case that you don't know, and I didn't mention before. And of course, all the coral reef degradation that we have seen for the last few decades, and we felt like we have to do something next. But this has to be combined with the restoration strategy. So we have corals dying, and we have all these different methods that we have been developing throughout the Caribbean and the world that is focused on asexual fragmentation for corals, having them in nurseries, the microfragmentation, and of course, the larval propagation. Next. So what we wanted to do is so for the stony coral tissue loss disease, what we have been doing is mainly applying amoxicillin, the antibiotic, to stop the disease. But we know that this has some issues, like it's not going to be as effective through time, and we can treat just so many corals. So we wanted to try to work and develop probiotics. That is something that is relatively new, and it's been evolving and getting more research, just developing probiotics, and especially working with 4 species that are important reef builders in the Caribbean, and are susceptible to stony coral tissue loss disease. So we're going to work with star coral, Orbicella fabulata, and Monstera cavernosa, and 2 brain corals, Diplora labyrinthiformis and Pseudodiploris tuberosa. What we want to do is evaluate the potential role of probiotics on the coral immunity, treat corals that have been affected or are affected by stony coral tissue loss disease, and compare those treatments to amoxicillin. It's going to be the same kind of time consuming, but hopefully in the long run, it's going to be more effective and less costly, because we know that applying amoxicillin with base to be is very expensive. And we want to see if the probiotics can help enhance the coral immunity, or at least resilience in healthy corals, and as well increase fecundity on adult corals. What we want to use the probiotics on coral restoration projects is involved with the use of microfragments and recruits in the different stages, mainly to see how they do in growth and survival. Next. So we're going to be working, as I said, in San Andres Island, and I'm sorry I didn't put them up. I'm going to apologize because I had a late invitation for the presentation, and then I was in the field and I got sick yesterday, so this was a last-minute presentation. But San Andres is located on the Western Caribbean. It belongs to Colombia, but is off the coast of Nicaragua. It has oceanic conditions there. and we're going to be working in 5 stations, all of them located on the western coast of San Andres. That is where the main reef is developed. What we're going to be doing is, so we have different phases. We're going to be doing stony coral tissue loss assessments and treatments, and we're going to do coral population assessments. We're going to do this by using photomosaics, and we have already set up some of these photomosaics on the island. We're going to be, just like I said, coral health and stony coral tissue loss disease, and we're going to use these plots as well for treatments. From year 1 to 3, we're going to use in some plots antibiotics, and in some other plots probiotics, and other plots that we're going to have as control. Next. So the metagenomic part is going to be combined with the probiotic development. I'm not the expert of this. I think Felicia was in this call, so if you have any questions, I'm sure that she will be able to answer those. She was with Sarah, and what we are going to be doing is collecting samples from the different species, at least 50 corals from each species, and just do the metagenomics. So with that, we can identify the bacterial strain or genes associated to resistance, and then develop the primers to target these biomarkers. And this her lab with Blake's lab, you're gonna be like working to identify the probiotics. Next. So the Yoshijima lab will be in charge of just developing all the probiotics candidates. So what we have done is collecting some samples, then we ship the samples. He will just start working on these samples and just choosing probiotic candidates. They will work with the metagenomic data that Sarah's provide, and then we can test the efficacy of these probiotics on corals in the field, and we can just like kind of go back and try more if it's necessary. And the probiotics are expected to be developed by next year, early next year. Next. So as I said, what we're gonna be doing with these probiotics is we're gonna try them in healthy corals to see how it works in terms of immunity, resistance, and as well as fecundity. So we're just gonna pack some corals and see if we have, after treatment in the different years, if we get any more eggs and sperm. We're gonna work with microfragments. Again, we want to see immunity resistance, but we want to see growth, and if there is any difference in the time that the microfragments take to heal the wounds when you cut them. And the last one is going to be with recruits. We're gonna see growth, and I'm not sure if we're gonna be able to see an immunity, but at least if we want to see if they grow better than some recruits that don't have any probiotics treatments into them. Next. So as I said, so far, what we have done is we have set up the permanent plots for monitoring. We have collected samples for metagenomics and probiotics, and we have started to do in the plots that we choose that we're gonna do treatments. We have treated the corals that are affected by stony coral tissue loss disease, so we have some data to compare to probiotics. Next. And I think that that's it. Thank you very much. And again, thank you for the invitation. And if you have any questions, just reach out.
Thank you ever so much, Valeria, especially for pressing on and joining us while you are really just getting over being quite sick. Our next CORDAP-funded project is establishing the Global Coral Cryopreservation Network, and it will be given by Anastasia Banaszak from UNAM in Mexico.
Thank you, Judy. Thank you for the invitation, and obviously thank you to CORDAP for funding the project, and to my wonderful companions in this particular project that all agreed to come together to get this project going. We've all been working pretty much independently, and now we'll be working together, and I think this will be a great advance for using cryopreservation in coral restoration and rescue. Can I have the next slide, please? Basically, we have 5 objectives in this project. One is to establish the core infrastructure that's needed for the global coral preservation network, which will include buying equipment that different labs like mine, for example, are lacking to really bring us up to date and be able to do preservation, not just of coral sperm, but also of coral larvae. And so this, I think, is a game changer in terms of coral cryopreservation. We also want to train the full team in larval cryopreservation and to implement this technology at all 5 project nodes. Excuse me, my phone's ringing. I'll just answer that very quickly. Sorry. Sorry about that, folks. So the idea is to basically make sure that at all 5 nodes we'll be able to do larval cryopreservation. And I think the reason this is a game changer is because it means that we may be able to accept larvae from other groups. So as long as the larvae produced and are sent to us or brought to us, we should be able to cryopreserve them in any given season. Then also as part of the sort of capacity building and training, we do want to produce an online cryopreservation system for practitioners to use, and also to build a unified database system for all coral biorepositories. We will then also conduct in-person cryopreservation training with new stakeholders. And at the moment, rather than sort of trying to get as many teams around the Caribbean trained in cryopreservation, we think the most efficient way to deal with this will probably be to have a couple of teams who are superbly well trained in these techniques and have them travel around the Caribbean and potentially around the world to do cryopreservation work in conjunction with the local teams. Next slide, please. So just to bring you all to the same sort of level on what cryopreservation is, it is the freezing and storing of live tissues and cells in an adequate medium at extremely low temperatures, like -176 degrees Celsius. So the idea is that although biological processes are halted, the cells are still alive and maintained intact. And so that way they avoid decomposition. And so this allows for maintenance over long periods of time, up to centuries. And so basically this is one of the techniques that we think that is really important in coral rescue and restoration. Next slide, please. So why cryopreservation? It's basically to maintain the viability and potential of sperm to fertilize. And that means that we can produce new genotypes through genetic recombination, through sexual reproduction, basically, using assisted fertilization. But we can do this over time and space. So we can basically collect in a particular season from colonies that perhaps are further away, and then be able to cross them with fresh eggs in a different year or in a different geographic space, right? So we could potentially, if there's a species that spawns twice a year or in successive months, that we can collect sperm, cryopreserve it, and then cross it with eggs from a different location in the next month. So this basically gives us a lot of possibility to increase the genetic diversity of the embryos that we produce. And also, potentially, we can also produce sexual recruits outside of mass spawning seasons, and that is in particular related to the larval cryopreservation. So it means that we could potentially thaw out larvae in the cooler months and then be able to settle them in the cooler months and have them out in the ocean and adapting to the reef environment. And obviously, cryopreservation in general is used to support restoration and conservation efforts of threatened species, and this is sort of the main reason why we are applying this to coral restoration as basically a plan B. So because these samples can stay maintained and live for decades, then as conditions improve on the reef, as we hope that they will in the future, then we do have another source of larvae and sperm to produce new corals once conditions have improved. Next slide, please. And so basically towards this, in terms of the work that we've been doing here in Mexico, we've established the Mexican Coral Gamete Biorepository. It's part of my lab in the National Autonomous University of Mexico, where I work. This biorepository started in 2016, and we so far have 6 species of reef-building corals. From a total of 77 spawning collections from 33 different reefs. We have 1,172 samples or accessions, as they're called, from 124 genotypes of corals. So basically, one of the things that we've been focusing on in this particular biorepository is to have as many species as possible, but also as well represented from different reefs, different depths and environmental conditions, so that we have that representation, genetic representation within our biorepository. With that, basically, I've finished. If there are any questions, I'd be more than happy to answer them.
Anya, thank you very much for a great talk. It gives me great hope that you're doing the cryopreservation work, and we're going to do all questions at the end. So we encourage everyone to stick around to the end so that you can ask questions and have a chance to chat with all the people who've presented so far. So it's really great. Thanks. Next, we're going to wrap up all of these by one more project that was funded by CORDAP, which is the Coral Restoration Roadmap project, with the goal of expanding coral restoration in coral reefs in Caribbean marine protected areas. And we've had the opportunity to team up with MPA Connect. We worked with MPA Connect for the last 10 years and have had a really great opportunity to work with them. And also with a great team of other scientists and experts and mentors who are working in the region. And they're listed here. You can see their logos below with Corallium, with Anya. And then we have Alize Zimmerman with Turks and Caicos Reef Fund. We have Stacey Williams with ICER Carib. And we also have Simon Walsh with Nature Island Dive. We have Elvita Chacron from Icamares. And we have Dana, who is with GCFI. Emma and Gabby are with MPA Connect. And we have Art Gleason with University of Miami, and Michelle Dennis with Institute of Agriculture at the University of Tennessee. So before we get started, I'm just going to, if we could, Tori, if you could turn your microphone off, just so we can hear some background noise, that would be great. Okay, so let's get started. So one of the things we've heard a lot about today is the impacts that corals have experienced over the last few years, from stony coral tissue loss disease to the die-off of diadema, and most recently with the 2023 coral bleaching event that occurred. We started to ask how can we start to address, to begin and rescue and recover these species, especially species that are at risk to stony coral tissue loss disease. Over 34 species have been affected. Many of these are species and families that are endemic to the Caribbean. They're highly susceptible to disease or bleaching. And we have species that are at risk of genetic loss and functional extinction. And many of these species are not normally included in current restoration activities. So that was something we wanted to focus on. And we also wanted to address how to expand coral recovery and ecosystem restoration in the Caribbean at the local level, but also at scales that are meaningful for recovery for these high-risk corals and to enhance ecosystem structure and function. So before we get started in meeting the rest of the team, I'd like to take a minute and have Dana introduce you to MPA Connect, which is an amazing network of MPAs throughout the Caribbean. Dana, would you like to join us here?
Sure, Trisha, can you hear me?
Yeah, welcome, Dana.
Okay, this will be super quick. Thanks for the opportunity. Hi, everyone. I'm Dana Wasenick-Mendez with NOAA's Coral Reef Conservation Program. And I just wanted to provide a brief overview of MPA Connect, which is, as Trisha said, a major partner in the CORDAP project we're here to talk about today. So MPA Connect is a partnership initiative between NOAA's Coral Reef Conservation Program and GCFI, or the Gulf and Caribbean Fisheries Institute. Many of you may have worked with members of our team. Emma Doyle is the coordinator of MPA Connect. And then also on our team, on our coordination team, are Gabby Ochoa, previously with Roatan Marine Park, as well as Christine O'Sullivan, based in Kingston, Jamaica. So as Trisha said, MPA Connect is a network of many MPAs. If you look on the map here, we work in 18 different countries and territories. We have a new dot on the map up here, and the Florida Keys National Marine Sanctuary is now a member of MPA Connect as well. But we work in all of these places in about 39 MPAs now to understand the needs of coral reef managers to support the effective management of their MPAs. And we facilitate a network among these MPA managers who share their experiences with each other and learn from each other's successes and challenges. And so we focus on a whole host of MPA management issues, from enforcement to fisheries management to how to do effective communications to biophysical monitoring. And most recently, we have added restoration, the restoration of coral reef ecosystems into our agenda. And so we assess the capacity of our MPA managers to do coral reef restoration, and then work to help them build their capacity to implement restoration programs. So we were really excited at the opportunity to partner with AGRA on this and to engage MPA managers in our network in this project to help them further along their coral reef restoration goals and objectives. So I think that that's it for me, but if anyone's interested in more information on MPA Connect, feel free to follow up with me or Emma Doyle or any member of our coordination team. Thanks, Trisha.
Thank you, Dana. And like you said, it's amazing to have these 39 MPAs spread across the Caribbean working together for the common goal of helping to protect our marine resources. And it was building on this that we decided to put in for a CORDAP grant, which we were really thankful that we've had the chance to get some support for, because this is going directly back out to these MPAs and others in the region. And I bring up this slide now because this is actually taken from a live satellite image, and it's showing just from 3 days ago how connected our ecosystem really is through currents. This is a current map, and if you go to the website, you can actually see it changes quite a bit, but you can see that we are connected underwater, but we're also connected on land too, through our different networks that we have, learning, sharing. And that's kind of the whole key to what we're doing with this CORDAP project is learning exchanges and trying to share that expertise. In collaboration with MPA Connect and all of the MPA Connect MPAs, we have gathered together a really great, what we call the Restoration Roadmap team. And this is a variety of scientists, on-the-ground experts, mentors who have worked in various parts of recovery and rescue and restoration. And you can see them here. I mentioned their names earlier, but it's really this team and with MPA Connect Network that we're excited to be able to work together. And as I mentioned, learning exchanges. This is one thing that we have heard from the managers who say, we really would like to get hands-on experience. We want to share our experiences. We want to learn from you. And this is just an example that MPA Connect had sponsored an in situ coral rescue workshop in underwater in the field training in Roatan with Roatan Marine Park. And we're really grateful for them. And started on what we call roadmaps. How do you actually begin to think about restoring? Where do you start? They have managers have a lot of other responsibilities, either with enforcement or managing communities and looking over fisheries. But how do you actually start to integrate the idea of rescue and restoration? And so we've been working with them to start to develop these coral restoration roadmaps and planning for that. So our main goals are to increase coral populations and improve reef ecosystem function, to help develop these science-based but management relevant. And that's a really key thing, to make them relevant for management, for how the managers can help implement this in their MPA, and to share and increase our technical expertise. Several of these MPAs have already been doing rescue and recovery, so they have a lot to share, but then also with our restoration team that we have to share with them other techniques as well. And one of our main focuses is really to synergize and kind of catalyze these restoration efforts in these MPAs. And if you look at in the background, the connection of those currents, we want to have that same connection through across these MPAs and start to try to expand restoration and scale up in the region. Our timeline for the next 3 years is for the first year is to help develop each of the MPAs, develop a portfolio of the data that they have, and then develop a priority for where they want to do rescue and recovery, what species, what techniques they might want to do, and then to really help them get that technical training if they need it, or to share their technical expertise. So that's kind of year one. Then we have a small grants program that we're going to help establish what we call legacy sites that will be, in these MPAs where they can start to do either expand the restoration that they have going on, or to start to just do coral rescue and recovery. And along the way, and you've heard this throughout all of the CORDAP projects, is really to communicate and help share this out with other people. So I'm really excited to learn and share from the other speakers today and from their projects, and then also with our MPA managers network. And then in year 3, we're going to continue with the restoration, evaluate, look at the progress, and then look at scaling it up across the MPAs so we can start to kind of connect the different MPAs together. And then in the last part is to evaluate and again look at next steps for continuing this, because there has been a great request to expand restoration, definitely needed in the region. Just this year, we have many training courses, which we're going to hear about in a few minutes, and opportunities to join in those online courses, and also to be able to have some sponsorships to do field courses, which we're going to hear about. And some of the focus that we're talking about is coral propagation. Anya is going to be talking about this in a minute, but this is really kind of thinking about how do we develop best practices for applying coral larval propagation to reef rescue and restoration. And it kind of starts off with just monitoring and where are corals spawning? And wouldn't it be great if all those 39 MPAs, if they were able to look at coral spawning so that they could help develop their own spawning calendars. And then also trying to look at larval propagation and assisted fertilization with the overall guidance of having best practices shared among these MPAs. Another big focus is ecosystem-based restoration, which Stacy Williams is leading through ICER in Puerto Rico. And this is looking at coral propagation techniques, both sexual and microfragmentation. There's a focus on stony coral tissue loss disease, corals that have been affected, but also looking at how do you design and develop and maintain land-based and in-situ nurseries, especially in areas where you may not have a lot of resources. How do you actually start? But then in addition to this, the ecosystem part is there's a lot of other things that go into the rescue and recovery, which is looking at enabling conditions, looking at herbivory, and Stacy's worked quite a bit on looking at sea urchin propagation and doing larval rearing in the lab, and has also started working with crabs as well. So it's learning how to do techniques to restore both corals and herbivores together, so you can have kind of an extra benefit of that. We put together what we call the Restoration Training Hub, where each of our Restoration Roadmap team members will be doing different courses. Again, they have an online portion that is open to everybody. And then there's also some opportunities to get field courses and hands-on experience with some of the Restoration Roadmap leaders. And I go back to this map because one of our goals is to advance coral reef recovery through collaboration and connectivity. And you see the different countries here in purple that are part of the MPA network, and you see the greens, those are the different sites. But it's kind of blank right now. And kind of our vision is if all of these MPAs have the opportunity to, a lot of them have data already, but if we can, start to connect them and so they can share, so that we can start thinking about recovery planning on larger scales. Also to think about, like, if we had a photo mosaic from each of these MPAs, we'd start to get a better picture of what is left out there and where those opportunities are. And if each of them were able to go out and do coral spawning observations, we would learn so much more about, which corals are still spawning and how they're doing. And then to think about ecosystem restoration at each of these local scales, but then kind of like your nearest neighbor, how can we help one another? So even though we have a lot of devastating events that have happened in the Caribbean, I'd hope for the future through this collaboration and sharing. So we do have these opportunities coming up, and we invite you to be part of them. And one is learning exchanges, to be a mentor or meet one of our mentors, technical trainings, both online and field courses, coral rescue planning, really kind of taking a step back from, looking at your MPA and all of the other things you have going on, and develop these action plans and share your expertise with others. And then looking at recovery potential. And we look at recovery planning at the local level, but how can we, look at it so that we can start thinking regionally? And then we have a small grants program to help do planning and implementation in the MPAs so that we can start to develop some of these legacy sites. So I am going to stop there. I'd like to thank all of our Restoration Roadmap team, which you can see their logos down below, and also to CORDAP for giving us this amazing opportunity. And I'm going to switch real quickly because we're going to take a minute, because I'd like to have our roadmap team talk a little bit about some of the courses that they're doing so that you can learn more. So if you are interested, we do have started a website where you can go. It's similar to our training websites that we have for our coral reef monitoring, but these are going to be based in the restoration side so that each of the instructors have a chance to share their resources and also their online materials. And the website is there. So first up, we're going to hear from Judy Lang, who's going to be talking about coral reef monitoring, and then to Art Gleason talking about large area imaging, those photomosaics that you saw earlier that Blairi had a chance to show, and then techniques in coral spawning, collection, and assisted fertilization by Anya. And Stacy will explain more about her ecosystem-based coral restoration of corals and herbivores. And Michelle Dennis will talk about coral disease and tissue sampling techniques. So next up is Judy.
Thank you, Patricia. Hello again. In the early 1990s, the carbonate geologist Bob Ginsberg asked if coral reefs in the whole Caribbean were degrading like the well-publicized and carefully monitored ones near marine labs or in protected areas, which question gave rise a few years later to the Atlantic and Gulf Rapid Reef Assessment Protocols. Next slide, please. The AGRA protocols allow standardized but rapid surveys of the key structural and functional attributes of tropical Western Atlantic reefs, like the corals and algae and fishes shown here, and fishery-independent estimates of fishing intensity. They were designed for sampling a large enough number of stratified reef sites to represent their average condition in any given area. Our in-country associates who've maintained or even expanded their monitoring efforts provide data of real importance on temporal changes whenever surveys can be repeated over time at the same sites. These protocols have been modified in response to the many changes affecting Caribbean reefs since 1998. We're currently on version 5, and we expect to make even more changes as photomosaics, drones, and other useful technologies become less costly and more widely available. Next slide, please. Now, for the Coral Restoration Roadmap Monitoring course. Its instructors are myself and AGRA's Patricia Kramer and Lynette Roth, plus Alize Zimmerman of the Turks and Caicos Reef Funds. So why monitor your project sites? Well, you should know what's there and the state of your reef's health, including that of its corals. You should also be tracking any changes that occur over time, in part to evaluate how your rescue efforts affect this reef compared to what happens at an untreated control site. Our course outline consists of 4 virtual modules that will focus on 1, sampling design and metrics for coral restoration roadmap monitoring. I think of these as the where, what, and why. 2, how to identify the common Caribbean corals and collect data that is relevant to your goal of enhancing viable coral populations at your outplant site. 3 refers to your reef ecosystem enhancement goals, which incorporate knowledge of A, the reef benthos, which is to be given first, and then B and C, reef fishes and reef structure, respectively, which will occur later this year. And then 4, data management and interpretation will comprise the restoration evaluation module. Next slide, please. AGRA has developed visual identification tools for fish, corals, other benthos, and for teaching the protocols. Anyone can now test their identification skills with some virtual practice quizzes, as shown on the right. During COVID, the classroom components of the training and the tests migrated online and have stayed there. The underwater components of training still consist of structured exercises with in-person instructors where trainees must learn to demonstrate proficiency during practice survey dives. And I can tell you that we all feel enriched when trainees from different areas come together to share information about their reef systems. And what we learn often improves our training and sometimes even modifies parts of a protocol. Next slide, please. So my AGRA colleagues have been very busy. They've now produced a new database for the current V5 protocols, a new online data entry tool shown on the top here that catches the many mistakes that are easily made while transcribing data, and a visualization tool giving surveyors access to dozens of graphical summaries of their own submitted data. And 3 examples are shown below. Plus, a highly sophisticated set of dashboards in a new Data Explorer now allow anyone to visualize all the processed V5 Caribbean area data surveys. If you haven't seen them, please explore for yourself sometime at the web address given on the slide and in the chat. And thanks for your attention.
Okay, I guess I'm up next. My name's Art Gleason. I'm a geophysicist working at the University of Miami in the physics department. I do a lot of different things related to imaging in the marine environment. Some of them are shown here. again, and I won't talk about these much because in the interest of time, the one that we're most concerned with for this talk and for this context is underwater photogrammetry. And so if you skip to the next slide, hopefully you see an example of what I'm talking about. Yes. So what do I mean by underwater photogrammetry? Well, what I do is develop and use tools to make 3D models of underwater landscapes like this one we see here. This is one of Lisa Carn's restoration sites from Laughing Bird Key in Belize. And in a lot of ways, these data speak for themselves, right? They're powerful visualization tools, powerful communication tools. And of course, you can measure quantitative metrics from this, things like benthic cover and relief and rugosity and so forth. So, and next slide. Okay, so this is just one other example showing a top-down view, 2D top-down view of a site at Chica Rocks in the Florida Keys. There's 2 time periods here. On the left is sort of peak bleaching during 2015, and on the right, post-bleaching, recovery or demise, as it may be. But the point is that the reason I wanted to show this was that these tools are really quite adept and quite powerful at doing fate tracking of individual colonies without having to do traditional techniques like tagging and all that. And you can really monitor quite a lot of colonies very effectively with this tool over time. So next slide, this is the last one for me. So I'll be leading a course related to this technology. It was something that I developed with some colleagues at the Scripps Institute of Oceanography and the NOAA Pacific Island Fisheries Science Center during the pandemic, and it's called Techniques in Large Area Imaging for Coral Reef Science and Monitoring. What we found over the years of doing this is that data collection is essential to this process. If you get good data, everything just kind of works, but if not, then it becomes quite challenging. So data collection and processing to make the mosaics is what this course will focus on. You can see a sort of outline of the topics on the right. And the basic structure is sort of a reverse learning approach where we have pre-recorded videos that go over the content. And then what we'll do is, so you can watch those at your on your own time, whenever is convenient for you. And then what we'll do is have sort of weekly or bi-weekly online kind of office hours where people can join and ask questions and share data, and we can discuss experiences. And it's proven to be a really pretty effective approach to getting this sort of data and tool out there. And I hope to see many of you join us. Thank you.
Thank you, Art. I am so excited about that. I say if there's one thing I could have done 30 years ago when I first started monitoring, I wish I would have been able to do photomosaics. Because they are just you can have a memory, but they are just great to be able to have as a baseline. So I'm really excited about your course, and we're going to have more information about how to learn more information about this course. But Art, really excited about that. Thanks so very much. Okay, up next we have Anya joining us back to talk about techniques in coral spawning collection. Anya.
Thanks so much, Patricia. I guess you can't get enough of me.
Nope. Always want more.
So one of the really neat things about this CORDAP grant is that we sort of have an idea that many different groups are really interested now in collecting coral spawning, assisting fertilization, and potentially even enculturing corals. And you've seen that there's a lot of advances, like from the Rita's group in Fundamar that Maria talked about today, as well as other groups like in Roatan, etc. That have, some of them have had training from us. And I think it's really great because we all learn from each other and then can build on our different experiences. So next slide, please. So basically, the components of this course is basically an introduction to the real basics and really basic methods of coral gamete collection and assisted fertilization. The idea being that this can work for groups that don't necessarily have culture facilities, which is probably the most of the groups around the Caribbean. But still, if you can predict when coral spawning will be and go out and collect gametes, you can actually assist fertilization and basically just release the embryos. And I think that's a big step, because I think one of the biggest issues that we're having in terms of production of coral larvae is the fact that because corals are now so much more dispersed, and that colonies of the same species may not actually be fertilizing in the natural environment, or at least it's probably really reduced, due to the huge percentage of colonies that we've lost between hurricanes, diseases, and bleaching, like happened last year. So I think even if we can just get out and just assist in the fertilization process, that would just be a really fantastic advance in getting more larvae out in the field without actually having to have culturing facilities. So basically what we'll go through is, and this course is both virtual and in person. The virtual part will be about 10 hours, where we'll talk about the coral life cycle and about reproduction and different methods in restoration. We'll also spend a fair bit of time on how to predict coral spawning, because that's the basis of all of this. The data for that are based on databases that we've had in Mexico since about 17 years now. And also with AGRA, we're looking at forming a Caribbean-wide centralized database for spawning information. And basically, we'll also talk about the preparation for field work, because the field work is at nighttime, so there's an added risk. So we definitely need to make sure everybody's really prepared. We're also talking about how to choose sites for monitoring spawning. And then for those who want to go beyond that and perhaps collect gametes, we'll be able to teach you how to make collecting nets and how to then monitor for spawning, how to collect the gametes, and all the tips and tricks that we have related to that, as well as assisting fertilization in the field, which you can do either on the boat or on the beach, and then also if you have lab facilities. Next slide, please. The next component will be advanced in-person training for those who do have facilities or the capacity to culture embryos and to settle larvae and culture coral settlers. That will be an in-person training, and it'll take 4 to 7 days depending on which species. That will be in Puerto Morelos in the summer. Next slide, please. So there are some prerequisites prior to starting, preferably a marine biology or science background or equivalent experience. We can basically decide on a case-by-case basis if somebody doesn't have that background, but has a lot of experience in the water, for example. Diving is not a prerequisite because we do have people who only snorkel or people who don't go out on the water, but are still very helpful in the lab. However, if you do want to dive, you'll have to be obviously certified for diving. You'll need to be medically cleared and be insured, preferably by DAN Insurance. Next. Prior to the virtual classes, we'll also be suggesting that people have a look at different videos or training course. For example, there's one in the Reef Resilience Network, which is a self-paced course that takes approximately 7 hours. But it's not a prerequisite. I mean, it's a suggestion that we would like for you to look at these videos or take the training course. I think that the webinars, it would be really great if you could have a look at them too. They're slightly less time. They're also self-spaced and really take about an hour to 2 each. And next slide, please. So for the people who are interested in doing the in-person training, the probable dates for spawning in the Mexican Caribbean I've outlined for these species. For Diploria, it's from July 31st to August 3rd. And for Acropora palmata, there should be a spawning in July and another in August. And for Acropora palmata, Fabulata, Annularis, and sort of Diploria strigosa, there's another set of dates, August 24 and 28. Now you probably realize that I've got two different sets of dates for Acropora palmata, and that's because we have different populations of palmata that spawn at different times, and they're quite separated. They're not very far separated by travel distance, but they seem to have very different spawning behavior in terms of the dates that they spawn. So while this is an added complication sometimes when we're monitoring for spawning, it actually is really great because we can go to different reefs at different times for collecting. And so this is one of the things that our spawning database has been able to show us, that populations on a particular reef will spawn at very different times to a population on another reef. We also will have a spawning for Annularis and Strigosa, as well as Sododiploria clivosa in September. So it really depends on who we pick for the different training sessions, because it will depend on what species you're particularly interested in and your level of knowledge and capacity to do larval culturing in your area. I think that's it for me. That's the last slide. Thank you, Patricia.
Thank you, Anya. I'm really excited about this course, especially the focus that you can do this work without having a huge lab. You can do it even if you just have a bucket. So it's depending upon what resources you have, I think she's able to, really teach you how to adapt for your region. Thank you, Anya. Next up, we have Stacy with the Institute for Socioecological Research. Stacy.
Hi, can you hear me?
Yep, you sound great.
Okay, perfect. Good afternoon, everyone. My name is Stacy Williams from the Institute for Socioecological Research, also known as ISER Caribe. We're a nonprofit organization here based in Puerto Rico, and we've been really focusing on ecosystem-based coral restoration. So for this project, we will be focusing on the theoretical and practical aspects of ecosystem-based restoration. The reason why we do this is I primarily started in restoration by culturing and releasing an important herbivore, Diadema antillarum, as you see on the screen and as you see in the picture on the lower left side that I'm holding. As you see, it's great seeing all the different projects improving coral survivorship on these reefs, because the reefs are changing rapidly. However, something is not changing, and it's the algal abundance on these reefs. So outplanting corals on the reefs with high algal abundance, these corals have a lower survivorship that we see here in Puerto Rico. So we've been currently outplanting corals and releasing herbivores to improve their survivorship. Can you go to the next slide? So Judy, I just went out last week to survey one of our restoration sites where we released Diadema. And this, in the beginning stages of our projects, we used to use corrals to keep the Diadema in one place in order to measure their herbivory. And as you can see, this is like in many coral reefs around the Caribbean. They're, covered in fleshy macroalgae like Dictyota. Here in Puerto Rico, we have Ramicrusta, which is an encrusting Paesionellid, and a lot of times these Ramicrusta grows under the Dictyota. That's, so it's just not Dictyota. And this is just a top-down view of the corral back in 2021 before we released any Diadema. Can you go to the next slide? A month after, you can, yeah, one before, yep, that one. A month after, you can see that the diadema did a really great job at clearing away this algae and creating a clean substrate. However, this is in a corral. So after the experiment, which lasted 2 months, we removed all the corrals. And like I said, I went back last week. Can you go to the next slide? And within 3 years, you can see that the diadema, they're doing a really great job in keeping the reef substrate clear of, there's still some fleshy macroalgae, but if you go to the next slide, if you look at, and then press, I guess it's on animation, you can see that in 2021 to 2024, still there is a low amount diadema, or actually was able to control this fleshy macroalgae in 3 years. And if you go to the next slide, and swimming all around this reef, you can see the impacts of diadema. There's more CCA, less fleshy macroalgae, and the corals look pretty healthy at this site. So restoration of herbivores is effective in maintaining algal cover very low. Go next. So with this course, we have an online course that will be giving some theoretical background on these different topics: ecosystem-based restoration, how to start an XC2 in situ nursery for corals, sea urchin, well, really herbivore restoration, because we are starting to culture the king crab. So we'll be going over methods on how to restore herbivores, and then coral microfragmentation. So these online courses will be a baseline, an introductory to get them ideas of what to expect during their visit and their 1 week course here in Puerto Rico. We will be giving them quizzes before and after. Also, like Art mentioned, I'll have an office time every week or every 2 weeks if necessary, if anybody has any questions about these different topics. At the end of July, we'll have them visit us here in Puerto Rico. And we have, if you go to the next slide, we have, we developed the Centro de Investigación Restauración de Organismos Marinos. That's in Spanish, but it's really just the Center for Research and Restoration of Marine Organisms. And we have 2 of these. These are the first, we developed the first land-based coral nursery and sea urchin and crab nurseries in Puerto Rico. We have 2 centers, one in La Parguera at the Department of Marine Science at the University of Puerto Rico, and another one in Ceiba on the East Coast. So participants will visit both locations, because both land-based nurseries are very different. And they'll be able to experience how to larval rear sea urchins, how to collect post-larval settlers of diadema, because there's different methodologies that we use to produce the herbivores, and also visiting our restoration sites and our in situ nurseries, and getting hands-on experience in the different topics of ecosystem-based restoration. So I think that is it for me. Thank you.
Thank you, Stacy. Your course sounds exciting. I love both the online and the virtual and the in-field person. And I love when we talked earlier that you're going to have a lot of your team who's actually in the field working and actually running these things are going to be helping you put together the course. And so it'd be great to hear their experiences. So I'm really excited to partner with your team and learn more. Thanks so much.
Thanks.
Next up, we have Michelle Dennis, who is with the University of Tennessee, the College of Veterinary Medicine. Michelle, she's going to talk about the diagnostic investigation of aquatic invertebrate diseases with focus on corals.
Hi, everybody. Can you hear me okay?
You sound great. Thanks.
Okay, great. So I'm Michelle, and I'm the oddball in the room because I'm a veterinarian. And so I spend a lot of my time teaching vet students and residents on how to diagnose disease, mostly in domestic animals, livestock, companion animals, but also wildlife and exotics. But in my research, I focus most of my most of my career has focused on disease in aquatic animals. So I started out in fish and then sea turtles and manatees, but my biggest passion has been aquatic invertebrates, corals, but also queen conch, urchins, spiny lobsters, and here in Tennessee, freshwater mussels. But the whole process is the same, and I'm going to come back to that in the next slide. So why am I here? I'm teaching this, or back on up for a second, just 1 second. I'm teaching this in the bottom right, the coral disease and tissue sampling techniques was kind of my specialty. And the reason why is because here I am in this photo showing me in my element, which is not in the sea, is actually like back in the lab with my slides and my samples. But I'm not the one on the front line. That's the resource managers. And so they need to understand this process that we use in other animals so that they can make the hard decisions that are coming at them fast. They need to be prepared. They need to know what's involved so that they can participate in this process. Okay, next slide. So that's what I'm going to be teaching through this course. And I see my involvement here in this project as a great example of applying a One Health approach. So this is where we get multiple disciplines together that aren't normally working with each other so that we can address these hard problems, these real challenges that are coming from stressed environments and affecting the animals that live within them. So the course title is saying that I'm going to focus on corals, but we're teaching you basic principles that apply across animal diseases, but also aquatic invertebrates. I have a lot of lessons learned that I'd like to share. And that too, kind of bringing the medical toolkit to address unusual species is also kind of fitting into this One Health approach. Okay, so button forward, and I'm going to bullet point some of the things that will be learned by those participating in this course. First, just how we respond to mortality events in any wildlife species. What are the steps involved, and what are those people in the field doing that's so crucial to this process? And I think that's on the next bullet point. So their observations really dictate what samples are collected and the steps that we take. So they are a crucial part of the team. And so we're going to get everybody trained up to be ready before there's an actual emergency to respond to, so that they know going in and feel familiar with the steps that are taken and how their observations are integral to this process. Okay, one forward. So part of this will involve teaching resource managers how to collect samples, which was initially what we were calling this section. You're going to collect samples, but it's way more than that. It's also collecting the information that's going to drive what we call the diagnostic investigation. So the samples without that information only gets us so far. So you will be able to collect samples, bullet forward. And then not just for pathology assessment, which is my bread and butter, that is kind of the central part of the investigation, but you'll have these tissues and there's a lot more that we can do with them, but they get processed in different ways and you're kind of out in a field environment, and what do you do? A lot to juggle. Sometimes you have to prioritize certain things over the other, so we'll work through how to make these decisions. Bullet forward. And then what happens to these precious samples that you spend so much of your time and resources going to collect? What does the lab do with them? And so that will help resource managers develop their expectations for this process. What do we get out of it immediately? What do we get down the line? And why we should continue to do it even when we're in circumstances where disease is outpacing the information that we're getting from our investigation. Okay, so, and finally, why is this so important to coral restoration? Well, so I said, I initially worked with fish, and, restoration is not new. It's not a coral specific strategy. We do restocking, propagation and restocking programs with fish. We've been doing that for a long time in lots of regions. Freshwater mussels, that's a big thing as well. And let me tell you that disease is an important issue. Whenever you start to propagate or farm animals, you are raising them in kind of stressed, unusual conditions where not all of their needs are being met. And this is kind of a nice recipe for emerging disease issues, maybe not right away, but down the track. And even in times of health, these propagation and restocking programs and other species, an important element of that is health surveillance. Before you start moving animals around, knowing what their health status is. And we use kind of the same principles and steps that you do during a disease investigation. And we heard Anderson Mayfield mention at the very start of this series, he said, we're going to start thinking a lot more about coral rescue and how we address that. And if we're going to have meaningful mitigations or means to support animals during a rescue process, we really need knowledge to inform that, to come up with good strategies. The more we know, the more creative and effective we can get. And that's where the steps in health and disease assessments become really crucial. So hopefully this course will empower resource managers in these ways. And that's what I have for you.